works with right now he's working with one particular strain or species of Clostridium, but he did his PhD on another species. So he's going to talk a little bit about that. And please feel free, this is going to be a pretty informal lecture, ask questions, okay? If you have a question or if you have a comment, that's going to make it really helpful for him and for me, okay? So I'm going to move to the back. And do you want one more light down?
Whatever, I'm fine.
Can you guys see it okay, or do you want me to try to put one light down? Lights down. Let's see if I can.
I'm going to reiterate what she said. Let's keep this very informal and just throw questions at me as they come. As Holly said, I work at the VA Medical Center in Boise, Idaho, the north end. I don't know if you've ever been there, but it's a beautiful campus. Most people don't even know it really exists. These are a couple pictures of it, an aerial view, along with a picture of our brand new research facility, $7 million, something like 20,000 square feet, and it's relatively empty. So we're looking for aspiring young investigators to come work with us. And so Holly asked me to talk today, and so I have some slides. It sounds like you guys are talking about the human microbiota and symbiosis, and then you're going to move into pathogens, bacterial pathogens, and that's what we do at the VA. Our group, an infectious diseases research group, work with many species that cause necrotic infections. I work with the Clostridium side of things. I've done most of my work with Clostridium sordelii, which you'll see is a very fascinating and quite bizarre infection. There's not a whole lot of money to do research on that organism, and so all the funding is available for difficile research because it's a problem around the world, and they're really in need of new therapeutic options to treat these infections. And so we've tried to work our way into that world to secure funding to keep things going with what we do there. So. And again, just ask questions at any time, please. I think it'll make this talk a lot more fun. So I'm not sure what you've discussed, but in terms of the human microbiota or microbiome, but as I'm sure you're familiar, our bodies are covered with trillions of these organisms. Okay, and that's because these guys were on Earth billions of years before we were, and so by the time we started to take foothold on this planet, we had to learn to live with these microorganisms immediately. And so through evolution, we've learned to live, again, in harmony with bacteria, viruses, or fungi that don't cause disease. And as you know, and as I'll show a slide here in a second, they're actually quite beneficial to us. The colonization happens at birth, and so they say most of your flora you acquire from your mother as you pass through the birth canal. And we have so many different microbes on our body that they say that we are really our own, each of us are our own ecosystem. So again, these bacteria are beneficial or are not harmful, and this is what you refer to as your normal or natural flora. And you're confined to certain parts of your body, and it's really taking up space or covering these areas that are exposed to the external environment, including your skin, your entire digestive system, vagina, nose, and throat. And again, we kind of live in harmony with these organisms, and it's literally a symbiosis. Of course, these two different organisms working together. And they help us. They help produce vitamins we're unable to synthesize, really help us digest food, boost our immune system, and then I think most importantly, help us protect us from disease. Okay, because what they do really, like I just mentioned, is they take up all these spots that potential pathogens could set up or shop or take over a niche to cause infection. Okay, and so not only do they provide this physical defensive barrier, but they also can remove pathogens from that may be residing or camping out in these areas by producing specific proteins like the defensins, these cationic peptides that can actually remove pathogens, or they create environments that are intolerable to certain pathogens, as you see here, that lactobacillus in the vagina. The stomach has a low pH, and there's Heliobacter pylori there, cause the low pH. But again, these type of environments can remove potential pathogens. And so it sounds like you're talking about this next week, but I just put this in here, and that's why there's such you read on Newsweek or you see on the news a huge controversy with antibiotics, and they're good and they're bad. You know, antibiotics, if you have an infection, let's look back 150 years ago, the Civil War, 200 years ago, people were dying from horrible Clostridium infections because you didn't have an antibiotic to treat these things. And so hopefully, or geez, you can think of anything. Pneumonia is another one. I mean, if you didn't have something to treat these infections, the patients were going to die. And so this is probably one of the most amazing inventions, if you will, or developments in humankind, is the development of antibiotics to treat infections. But when you overuse these things, which is what's happening today, people go in because they have small ailments, you know, even a scratch on your skin, you don't feel well, people want antibiotics, and they'll rummage through people's cupboards and steal these things. But what you're doing now is you're really altering, well, not only your own body, but you're altering these pathogens themselves, because you're abolishing now, overuse of antibiotics, you can abolish your normal flora, because those too are bacteria, and by doing so, you're now providing opportunity for pathogens such as the one I work with, Clostridium difficile, leads to yeast infections in women. And actually, I have in the bottom here, I saw a really very interesting study at a conference. Every time you take antibiotics, you change the populations of your flora. And so this professor was looking at the gut populations, and he had a profile of several individuals, and then after they took antibiotics, they looked at the populations again, and they're completely different. You kind of recolonize yourself every time you expose yourself to antibiotics. So I thought that was fascinating. And so, people claim, I have an uncle that claims that, he was never lactose intolerant until he was on heavy antibiotics, and now he can't process lactose. And I thought, you're crazy. But now, after going to that study, I thought, well, maybe that's what's going on. He actually switched his actual flora to where now he can't break down lactose. So
I guess where's the breakdown then if you have an infection of some sort? I mean, is it within an infection, like a bacterial infection, then yes, you need an antibiotic, or is there a way that,
it's a risk-reward thing. I mean,
your body can naturally kind of get rid of some of that?
Me personally, I haven't had antibiotics in probably 15 years. They scare me, especially with all the work that I do with this difficile. It's opportunistic. I had a really bad sinus infection, and I went in and they said, well, here, take this antibiotic. And I'm like, oh, screw it. I'm just going to try to fight this thing off, because I didn't want to wipe out my flora. So it's kind of the risk-reward. Now, if I have gangrene, or if I have something, well, I'm taking an antibiotic immediately, because I don't want to lose a limb or something nasty. But for me, if it's a cough or even something on your skin, or try to fight that naturally, I'd use maybe a topical, your what is it called, your Neosporin, something like that. But I'm not gonna take ciprofloxacin for 2 weeks, because it's gonna wipe me out. And then again, with what I know, I'm colonized with certain pathogens, and we were just talking about this. Hopefully that means I'm developing immunity, so when I'm old, I can fight these things, older. Yeah.
So since you're in the laboratory working with this, how close are they to finding something to cure MRSA and CDIM? Because I know it's resistant, obviously.
Yeah, that's the problem with these two, and we'll talk about that, and she'll talk in further lectures, but they're developing resistance because that's what happens. You keep exposing these things, these bacteria, to all sorts of antibiotics over time. They're amazing little creatures, bacteria. Remember, you can, in one piece of stool, there's more bacteria than there is the entire population on Earth. And so they reproduce every 20 minutes. And so, I mean, they're amazing. They figure out ways. Genetic replication is not perfect. And so every time they replicate, there's a potential mutation that takes place. And these mutations are what lead to, to new ways to become resistant to these antibiotics. And so you'll expose culture of bacteria to antibiotics, and it's wiping them out, and it's wiping them out. All of a sudden, something will start to grow, and you'll be like, well, that's weird. I put the antibiotic in there. And over time, this doesn't happen overnight, but one of them will have the right mutation that develops a system that will either cleave or clip the antibiotics, or pump them out of their own systems. And so they develop their own. They're very smart, and so it's kind of the evolution, the cat-mouse game. We develop a new way to treat an infection, and then they develop resistance, and then we develop another strategy, and they develop resistance, and it's going to go on forever. And so that's what you read in Newsweek, that there is so much money for research to develop new antibiotics, because we've overused them so much over the last 50 years that we're kind of running out of options now to treat several infections. This one, big time, difficile. MRSA, does everybody know what MRSA stands for? Methicillin-resistant staph aureus. So it's Staphylococcus aureus is the pathogen here. And so for decades, people used methicillin to treat these staph aureus infections. It's a very common wound infection that you can pick up contamination. And so it came out, the press does a good job of really making these things right, making these things sound much worse than they are. But they developed resistance because we exposed them to methicillin for so long that now there's methicillin-resistant staph aureus, so you hear that MRSA. And so, I've seen people used to work in a hospital. Some woman found out she had MRSA, and she absolutely flipped out in this lobby, and had to console her. And I'm like, it's not, the press makes it sound like a monster, but it's like, oh, maybe there's other organisms that have developed resistance to literally 12 antibiotics, like difficile, where this is just, in this case, one antibiotic. And so it's a lot worse than that. But my point, nonetheless, is that expose them long enough, you start to develop resistance, and the overuse now has created kind of headaches for physicians. Go ahead.
I was going to say, one other thing that I read was that the pharmaceutical companies aren't really going after this as much because they're focused on broad spectrum, which makes them a lot more money than the specifics of
It
that.
depends. Well, yeah, it depends. In this case, difficile is a problem around the world. It's not a problem in Idaho or the United States. It's a problem in hospitals globally. And so if you could come up with a new antibiotic to treat these things, you'll be on a beach somewhere because the whole world is going to need this. And so there's a lot of, we do a lot of testing of new antibiotics. That's a lot of my research funding now is, I'll show you the study later in the talk here, but basically we show that some antibiotics actually trigger these organisms to produce more toxin. And so these companies kind of come to us and say, does my antibiotic do that? And so we do this work to show either that it does, or typically they don't know they're new antibiotics because these bugs haven't seen that yet. I think there's research for both specific and for broad spectrum.
So with C. difficile, one of the treatments is the fecal transplant.
Yeah.
That they do.
Yeah.
And I was reading about, is that illegal in the United States?
I don't think it's legal. We'll get to that. That's later in the talk, but no, I was telling Holly too.
So is the idea then to get away from antibiotics and try to find alternative
Well,
ways
for
to
that, these bugs that I work with, Clostridium, they're spore formers. And so, in theory, these things can lay dormant in a spore forever. In fact, you could take a shovel, we could go out right out here in the backyard and probably find anthrax. You can find all these Clostridium spores in the dirt. They're soil dwellers, and they wait until the opportunity is right, and then they germinate and cause infection. And so, you can treat difficile infections with some antibiotics, and it will get rid of the bugs. But the antibiotics treat the bugs, not the spores. And then once things happen and the opportunity is right, these spores germinate again, and then you have the infection again. And so it's hard to clear out the spores. So if you can wipe everything out and reintroduce, new flora and try to push everything clean, if you will, and then recolonize with good bacteria that are absent of these spores, it seems to be beneficial. We were talking, I think it was an argument 10 years ago whether or not it was actually helping. Now I think it's accepted that it's a good therapeutic benefit or alternative for some patients. So good questions, this is great. So, okay, so now you're learning, okay, well then there's good and there's bad bacteria, okay? And so, I'd say most infections, many infections that we get are from good bacteria getting in places that they shouldn't be. Urinary tract infections or pink eye or even skin wounds, you're getting normal flora in an environment where they really shouldn't be, and so that's what's causing the infection, okay? Take antibiotics if you want, or for me, again, I would rather use the Neosporin to get rid of them, or get rid of that infection, kind of get them out of there, and then you heal up. But there are a handful of bacteria that we call the bad bacteria that are pathogenic microorganisms, and they're pathogenic because they produce exotoxins, and that's what makes them so nasty, all right? That's what's causing these horrible infections. And they're just proteins secreted by these bugs that really have developed over millions of years. Again, these bacteria were on Earth, 1 billion years before before eukaryotic, cells or organisms, multi-celled organisms started to take foothold on Earth. And so then these bugs had to develop ways to compete with the other bacteria for certain, environments or niches. And so I think with millions and millions of years of evolution, and as organisms like ourselves started to arrive, these toxins then kind of evolved to then take over these different organisms that were walking around Earth to compete with them for whatever it is, nutrients or things like that. And so it's now to a point where, again, a handful of bacteria can cause horrible infections in humans. And so that's what we see here. I think this is measles, and that's smallpox, chickenpox. We have anthrax. Of course, we all know what HIV is. That's botulism and tetanus, and that's leprosy, and it goes on. I could show about 15 more slides of examples of bacteria causing or producing these exotoxins that have these horrible ramifications on humans. Have questions on that? So that's what makes them bad. And so that's where you definitely want to use antibiotics if you were to. And back then, like leprosy, I don't know if you guys ever watched the movie Braveheart, for example. What is it, the Prince or the King of Scotland? He's dying because he has leprosy and there's no antibiotics, and hopefully you either fight it or you don't. And there was no option back then again. So it's very critical, and it's really pushed we as species forward, having the ability to fight these horrible things. So this is where you want to use them. Okay. So with that, that's kind of my introduction now into really what we do. And so kind of what we say in all the grants that we write is we're really trying to find new ways to treat these infections, because Clostridium infections are so often so rapid and so quick, and they produce not one toxin or two. Most of the time they produce several toxins. And so if you can't treat these things immediately, chances are you're going to, well, these days you probably won't die, but you're definitely going to lose a limb, something like that. And so we're trying to come up with novel ways to treat these very aggressive infections. And so the first one I'll start with is Clostridium sordellii. And as Holly alluded to, was my PhD thesis when I started the VA years ago. And when I got there, you're probably like me, I never heard of the organism before. I heard of Clostridium perfringens, but never sordellii. And so it's very rare. We put out an article, a review article, and I found 50 documented cases of sordellii since 1910 or something like that. Very rare. And so my boss said, you should do your project on this because you're not going to be competing with people around the world to get the data. You can take your time. And I thought, well, this is great. So I'm a type B personality, so that was great. Perfect. But I was working on it for a couple of years, and all of a sudden all these headlines started coming out in newspapers around the world, and women were dying specifically from sordellii following the use of the overnight abortion pill. And so nobody knew why. In fact, we got contacted from the federal government. They knew that my boss was working with it, so I was 28. And I got to fly out to the CDC with my boss, who gave a talk at the CDC, and I got to sit in the crowd and watch. It was really cool. But it kind of made this, it was a little splash in the infectious disease world. It drug it into the spotlight, if you will. And so we got some money to study it. The fire has since died down, and there's not the money available that there was. But again, it's because, too, it's a very fascinating infection, okay? And it causes these myonecrotic or gangrene-like infections in a variety of animals, cattle, sheep, and we as humans. In fact, we had a giraffe at the Boise Zoo 5 years ago die from a sordellii infection, okay? And so most infections are associated with young, healthy women giving natural childbirth. However, they've now moved into these bizarre infections involving the overnight abortion pill, and actually there are 3 outbreaks, sordellii outbreaks, published that happened in the state of California from IV or skin popping drug users. They were taking their heroin and they were mixing it with dirt, and then these poor souls were injecting dirt into their system and were coming down with these sordellii infections. Horribly. Yes?
Okay, so I did see the giraffe that had a hurt leg at the zoo, and I was upset about that.
I did not do that. I did not.
Okay, I was upset about that because because,